Okay. Good afternoon, everyone. Welcome to this lunchtime session on plant tissue culture of crops. I'm just going to share my presentation with you. You seeing that? Okay, again, good afternoon. Welcome to the session on plant tissue culture of crops. My name is Farah Aligour. I'm the Biochemist II at the Plant Tissue Culture Laboratory at Research Division, Ministry of Agriculture, Land and Fisheries. We're located at Centeno. That's just behind the old airport. I have with me my colleague, Mr. Deodat Sigoven, who will be helping me to collate some questions at the end of the presentation. So I think we'll hold out with any of your questions till the end. Today I just want to share with you a little bit about what I do and the basics of plant tissue culture. So first of all, we'll be looking at the principles of this technique. What is it all about? What is plant tissue culture? As well as the steps involved, the technique of the tool that is used, and we'll look at some applications with examples. And finally, we'll put it all into context by looking at the importance for national agriculture. So what is plant tissue culture? Basically, plant tissue culture is growing of plants in a jar without soil. So small pieces, any part of a plant can be used. So for example, a piece of a root, stem, leaves, any plant in theory could be an explant, which is a piece of plant used for tissue culture. It is placed on a synthetic medium, which is this gel-like substance that you can see in the picture on your right-hand side. This synthetic medium has everything that the plant needs to grow. It is artificial, and we usually use something called agar to make it gel-like so that it provides support to the plant. All of this is done under sterile conditions in a controlled environment of temperature, humidity, and photoperiod, which is really the daylight interaction. So we put on the light for a certain amount of time and we take it off to mimic the day and night period that a plant usually experiences. Unlike traditional propagation, this is done in a very small space. I want to introduce a concept called totipotency. This is the reason why tissue culture is possible. This concept states that it is the genetic potential of a plant cell to produce the entire plant. So let me break this down. Every plant cell has the genetic material within that cell to create an entire plant. What makes a cell, a root, a shoot, or a stem for that matter is a process called differentiation. So differentiation will determine what parts of the plant does that cell reside in. Plant tissue culture, it allows de-differentiation of your cells within the culture. And then these cells are reprogrammed to become shoots, roots, and eventually into an entire plant. So this mass of de-differentiated or undifferentiated cells form something called callus. So I'm not sure if you can see this. This is the callus within a test tube. This is the mass of undifferentiated cells. Is a lot like stem cells in animal cells, where they have no purpose at that time. So moving on to the technique. There are 4 stages to plant tissue culture, 5 if you count explant selection. So these stages are stage 0, where you take a piece or any part of the plant that you wish to culture. Then you establish it aseptically in culture, which really means it's free from microorganisms. Then you induce the formation of shoots. In vitro multiplication of shoots means that you have in vitro literally means in glass, is a Latin word. So it's just like you have a shoot in a jar, whatever container that you have, and you cut it up into small pieces, and each of those pieces could become a new plant. So that's stage 2. Stage 3 is in vitro rooting, where each of those explants are induced to root by the use of plant hormones, which we'll talk about a little bit later. And finally, transplantation and hardening of the plant before field establishment. Let's dive a little bit into each stage. So stage 0, explant selection. Some people leave out this stage, but I think it's very important to select a healthy plant at the very beginning. Just like any method of propagation, what you get at the end is it really depends on what you start with. So as mentioned before, any part of the plant can be used, but in terms of selecting a plant, you look for a healthy, disease-free plant. Disease-free and free from water stress as well as any nutrient deficiency. So when you go out into the field, you select the best looking plant. A number of factors should also be considered in selecting your explant for plant tissue culture. Age is one of those. So younger explants tend to respond better in culture than older ones. Season, the weather conditions as well has a big role to play with. We tend to collect in the dry season because we get less contamination around that time, as well as if we are culturing buds and shoots, then we choose the season where they are flushing. So at that time, they respond better in culture. The size of explant should also be considered. Larger explants tend to respond better in culture because they have a reserve of nutrients that could propel their growth in culture. And obviously the species some species grow better in culture than others. Some are even very difficult to culture. What you're seeing in the pictures is embryo culture of coconuts. It's one of the research projects that I've been working on, where a 10-month-old nut is collected. The embryo is excised and cultured in vitro. So that's one of the research activities I'm involved in right now. Moving on to stage 1, establishment of aseptic culture. As mentioned before, we have a media that we place the explant into. Now, this media is rich in nutrients and vitamins. Any growers in the audience might recognize the elements N, P, K, nitrogen, phosphorus, potassium, which are macronutrients, and these are also in the media. So we have calcium, magnesium, sulfur, and iron as well. Well, unlike traditional growing of plants in the natural environment, we must supply the plants with organics like carbon, hydrogen, and oxygen in culture. And minor elements are also important in culture, such as boron, cobalt, copper, and iodine. Because this media is so rich, it also encourages the growth of microorganisms. And if we have microorganisms growing in the media, it defeats the whole purpose of trying to grow an explant in culture. So that is why theme throughout tissue culture is keeping everything sterile. And two pieces of equipment that are very important in plant tissue culture to maintain the sterility as the autoclave, as well as your laminar flow hood cabinet. The autoclave is used for sterilization of instruments and media using steam. It's much like the pressure cooker you have at home in your kitchen. This steam kills out all the microorganisms. The laminar flow hood, it creates like a sterile workstation for any transfer of explant or plant material that has to be done. The surrounding air is pulled into the cabinet and pushed through a HEPA filter, and sterile air is blown across towards the operator. I'll show you something, a video of it in a little while. So here we have a video of establishment of banana interculture. This video is showing Mr. Terrence Jack, who is a colleague of mine. He was very instrumental in helping me to set up this lab. Here he's doing sterilization procedure for banana culture. Mr. Ramdas, are you seeing the video playing? Okay, thanks. Right, so here we have Mr. Jack doing the final stage of banana sterilization. We did bleach and alcohol before. So what you see in here is really the meristem tip of the banana with part of the pseudostem as well as the comb. So we are doing 3 final rinses in sterile distilled water. You can see the laminar flow hood in the background. This next step is removal of the peeling back of the pseudostem. These tissues would have been damaged by the sterilization procedure. So the final product that you want, it would be healthy tissue free from any microorganisms. So this is a very delicate process, and we continue the peeling back of banana with the pseudostem, but the final product will contain a piece of the pseudostem as well as a piece of the comb, and then it is initiated into culture as shown here. Okay, so we have a gel-like medium, and we gently push it into the medium. So this is a demonstration of establishment into culture. Before I move on to the next stage, I just wanted to mention a little note about plant growth regulators, because it is so important for plant tissue culture. As before, I explained the nutrient content of the media for plant tissue culture. But plant growth regulators are produced naturally in the plants, and it aids in developmental processes within for plant growth. So, for example, plant growth regulators, they control processes like senescence, which is when your leaf dies off and drops off of the plant. It controls processes like phototropism, that's movement of the plant towards light. But in plant tissue culture, the major hormones that are important are two, auxins and cytokinins. Cytokinins play a role in development of shoots, and auxins is very important for development of roots. However, not only their absolute concentrations matter, their concentrations relative to each other also matter. So if you have a media, as shown on your right-hand side here, with equal amounts of cytokinin and auxins, you get formation of callus tissue like this. Callus tissue like this. And from this callus tissue, if you apply a high concentration of cytokinins, you get the formation of shoots. So I'm not sure if you can see this tiny shoot here forming from a callus. Moving on to stage 2, in vitro multiplication of shoots. This stage is really where the beauty of tissue culture is. Here we can have mass multiplication of shoots in vitro at a faster rate than what can be done conventionally. So this is again is a sterile technique done in the transfer room. And so if you have a single plant that can give you a minimum of two plantlets in vitro, then from those 2 you can get 4, and from those 4 you can get 8 and 16 and so on until if you look at the bottom here, you have what is called exponential growth of shoots. Multiplication of shoots is an exponential process where in a couple of cycles you can get millions of plants. So a lot of people commercially take advantage of this aspect of plant tissue culture. So I'm not sure if you would have heard of micropropagation. Micropropagation is a component of plant tissue culture that commercial growers take advantage of. They do this for plants, like especially high value ornamental species like orchids. I know all of the strawberries that are produced now are also from tissue culture. So I just want to move on to another video on in vitro multiplication of sweet potato. This is just to demonstrate the technique for multiplication. So this is me in the laminar flow hood. Usually I use alcohol lamp for sterilization of all the instruments at the very beginning of in vitro multiplication, but these days I'm being forced to use something called a glass bead sterilizer, which is another piece of equipment because alcohol has become so expensive during the COVID period. I mean, we're all trying to adapt, right? So here I'm sterilizing each piece of equipment. This process, it takes a lot of patience. So here I have a sweet potato explant. It's first going to be defoliated and then cut up into nodal cuttings. So first I take off all the leaves and then I cut off each node that can be salvaged. Plant tissue culture is different from macropropagation because it is so small. You have to remember the orientation of the plant. So what I usually do is I make the bottom part under the node a little longer than the top. So I know which part has to go into the media. And so from this single explant, you see how many nodes I'm getting there? And then each is placed into a new tube and cultured again. So from this single explant, I got about four explants. Okay, moving on to stage 3, in vitro rooting. So once you have development of shoots in culture, we induce the formation of roots by applying hormones. Basically, we apply auxins in culture, in vitro or ex vitro, which is out of culture. For example, rooting can be done in perlite for some crops. Stage 4, transplantation and hardening, is when you have a fully developed explant like this one. You have a fully developed explant with enough shoots and a proper rooting system. You can now take this out of the agar, wash out the roots very carefully because they are very delicate. And this is placed in humidity chambers in the greenhouse with low lighting. It's important to treat these plants very delicately, like babies actually, because they have been growing in culture all their life. They have been getting all the sucrose they need, getting all the nutrients they need in high humidity. So at this stage, actually, people get the most losses when doing plant tissue culture, because this transition into a new environment is not always successful. So eventually, when you keep your plants in the greenhouse, high humidity, low lighting, eventually lower humidity and increase lighting so that the stomata can develop properly, because they won't have been fully developed in culture, as well as the plant can become autotrophic. That means that it produces sugars on its own. It undergoes photosynthesis. There was no need for plants to undergo photosynthesis in culture. So the plant has to learn to survive in this external environment. Once you have success at this stage, then your plant tissue culture process is over, and then you're ready for field establishment. Here is transplantation and hardening of some cassava varieties that we have done in the past. So that was the technique of plant tissue culture. Moving on to applications of plant tissue culture. Plant tissue culture is used as a tool to achieve many objectives. So one of those objectives are commercial micropropagation. There are millions of plants being produced annually by plant tissue culture on an international scale. Mainly in the past 40 years or so, plant tissue culture has become a big industry for orchids, as well as they're doing plant tissue culture in bananas. Bananas was one of the first crops to be plant tissue cultured, as well as potatoes. And recently, cannabis tissue culture is coming on board. I'm hearing a lot about the plant tissue culture of cannabis for its propagation. There are even biofactories. So the picture that you're seeing on your right-hand side, with this researcher holding up some coconuts. So that's coconut embryo culture in jars. What they do in Mexico is that they do a process called somatic embryogenesis, where they induce a normal cell, not an embryo, to become an entire new plant. So that is being done on an international scale. Plant tissue culture can also be used for germplasm conservation to protect against loss of biodiversity. The advantage of doing this is because so say for example, we have a natural disaster or a disease that wipes out a species. We can keep plants in a sterile medium within an indoor environment that can be regenerated and put back out into the field after such a disaster. It can also be used for screening cells for advantageous characteristics in plant breeding. So traditional plant breeding would have the plant breeders will have to wait until plant reaches a certain size to see a particular characteristic like disease resistant or flower color or whatever phenotypic expression that they're looking for. But by detecting biomolecules or certain key things within the culture, this process can be speed up. So that plant breeders use plant tissue culture as well. Embryo rescue is another application of plant tissue culture whereby immature or weak embryos are made into a viable plant by controlling the conditions. It's also used to produce clean planting material from virus stock. If you have a field of disease plants, you can take the apical meristem tip from any plant within that field and culture it, and it's supposed to be disease-free. So you can get a disease-free, clean plant within one generation by using this technique. This is because the meristem tip, it multiplies so quickly that it is virtually impenetrable to any viruses, bacteria, fungi. So, but this takes a little while to do for your crop. And also, plant tissue culture is used in the safe movement of germplasm, which is one of the key activities of the lab that I work in. Just a special mention about the cassava industry development project that I was involved in, I am involved in fact. So this was a collaboration with the Food and Agriculture Organization of the United Nations for the development of the cassava industry. Here, the plant tissue culture lab played an important role in the importation of 3 superior varieties of cassava from CIAT, which is an institution in Colombia that has a lot of varieties, and they keep a germplasm bank for cassava. So these varieties were imported via tissue culture. So we got tubes containing MX59, MP 183, these kind of varieties, and we multiplied them in vitro. And we were able to put them out into the field, and now we are in the process of doing the first evaluation trials for these crops. Under the same collaboration, the laboratory has also prepared 4 local varieties, 4 important local varieties for farmers to be sent to CIAT for identification of their characteristics as well as for DNA profiling. And once this DNA profiling is done, this should be the start of a germplasm bank that we have all the characteristics, and we call it passport data for a crop at Centennial, at Research Division in Centennial. So we hope that that comes about. Also, when these plantlets are regenerated, they'll be sent back to us so that I will have clean, disease-free material to propagate and possibly for distribution. So Plant tissue culture is very important for national agriculture. As mentioned before, we have a problem when plant material is transported across borders. So if you take plant material from one country and bring it into this country, then you risk bringing in pests into the country. So what international organizations has come up with are safe guidelines, guidelines for the safe movement of germplasm. And one thing throughout all these documents is the use of plant tissue culture for international exchange of germplasm. So that's one of the key activities of my lab, is to facilitate the international exchange of germplasm, which I have done with cassava already. And another importance for our agriculture is conservation of national genetic resources. I've started the conservation of cassava and sweet potato, and in the future, I hope to conserve much more crops via plant tissue culture. And I spoke about virus elimination and disease-free micropropagation. This is where we can, so if we have a disease that is plaguing a particular crop, research can be done to eliminate viruses within that particular crop and produce, again, disease-free material for use by farmers. So I just want to leave you with a few pictures of citrus production in Florida. This disease, HLB, hornlungbing or citrus greening disease, is very rampant and prolific in Florida. Some people may know that the citrus production is very big there, and they have transitioned into the use of plant tissue culture for all their planting material to fight against this disease. Yeah, and that's it. I hope you enjoy my presentation. Any queries or questions after the session, you can send to tocropsces@gmail.com. So this is the end, and I'll take your questions now. There are no questions as yet. Feel free to ask me anything. I have my colleague here who can help me with answering any question that you may have. Okay. Okay, so I have a first question here. Can this technology be used by farmers, or do we have to go to a lab? This technology, it can be done at home with modification of certain places in the home. For example, the preparation of media can be done in a kitchen setting. If I am to say what are the two most important pieces of equipment that you need, is an autoclave and a laminar flow hood. I know of some persons who have tried it at home with some success. It's just that you need to have a setup and dedication to this technique. It must be beneficial to you. So it may not be that cheap, but if you're getting profits by mass propagating a certain plant, it's definitely a way to go because of the production of disease-free planting material. What are the limitations on the public accessing these services? You want to take that one?
Right, so our laboratory is a research facility, and as such, we conduct research on developing protocols, etc. We're not into production or mass production. The only space I know is persons have been able to access tissue cultured bananas, I believe plantains from truly Tobago House of Assembly. I don't know if they produce it themselves or if these were imported. But so our facility is not into production to give out to farmers, etc. So we're more or less into the research aspect of it.
Okay, and the other question is, how can farmers be trained in the use of tissue culture? I know of a course that was being given at the University of the West Indies on plant tissue culture for home and semi-commercial production. So I would advise that you can contact University of the West Indies for formal training in this method.
Are there YouTube videos?
Yeah, definitely. You can get a lot of information on YouTube as well as Google on this technique. Any other questions? Can you just repeat the solution that is used for the process? I don't think I understand that question.
I think you talk about the media.
The media? Okay. So if you are speaking about media preparation, the solution or the media is you can get it pre-formulated. It was developed by two people, Mirashig and Skoog. So they call it MS Media. And you can buy this from any scientific company in Trinidad, Southern Systems, Western Scientific, any of these scientific supply companies, and they can sell you it pre-packaged, and you just have to add it and put in your agar as well as adjust the pH of the solution, and then you can have your media ready for plant tissue culture. But I must mention that each crop will have a different protocol that is required for its growth. So once you can get the protocol for the particular crop, then you can now start to manipulate and optimize the procedure for that crop. Some species are more difficult to grow in culture than others, and it is beneficial for some species, and some species is recalcitrant. It's very difficult to culture some species. So,
right. I'd just like to say that tissue culture is not limited to only food crops. It's across the board with respect to all plants. So, for example, I know of local nursery owners who actually import tissue cultured orchids into Trinidad and Tobago. So they would import it, harden it, and sell it to the public. So it's throughout, right? Both food crops and ornamentals, etc.
any more questions? Okay, I'd just like to thank everyone for joining me. I hope you had an interesting lunchtime, and feel free to contact my technical officer at tocropces@gmail.com for any other queries and questions that you may have.
We'll try our very best to answer them.
Okay, thanks everybody.